What's up, everybody? Mr. Lane's back, and now today we are talking about the brother who is the father of modern genetics. That's right, the one, the only, Gregor Mendel. That's this old dead white guy that we're looking at on the board here. So Gregor Mendel, he is known as the father of modern genetics. But I also called him a brother because he was actually a monk who lived in Eastern Europe in the mid-1800s. And because he was a monk, he had a lot of time on his hands. And what he did is he liked to grow peas. He actually was not his first choice. He was actually a substitute science teacher in high school, but he really kind of sucked at that job. So he got booted out into the garden and he just started kind of growing peas. So serendipitously, he ended up becoming the father of modern genetics, but he unfortunately never knew that. So Mendel and his peas. First off, genetics, it's the study of heredity, which means how traits are passed between generations. Gregor Mendel was an Austrian monk whose work was important to understanding heredity. In fact, he was the first one to say that traits are passed between generations. What's really wicked cool here is that he was actually a contemporary of Charles Darwin. Imagine if Darwin and Mendel had gotten together and talked about how information is passed between generations. It would have been like mind-blowing. But unfortunately, they never met. So Mendel carried out his work with ordinary garden peas. And we are not talking about like a couple of peas here. We're talking about 27,000 individual plants over the span of several years. That is a lot of peas. So, I mean, I don't know about you, but I think he was really kind of an advocate for peace and prosperity. In fact, you could say that he believed in world peace. During sexual reproduction, sperm and egg cells join together through the process of fertilization to create something called a zygote. Pea flowers are self-pollinating, which means they have both male and female parts. That means the egg and the sperm from the same plant can combine to make another pea. A trait is any specific characteristic that varies from one individual to another. Typically, we think of traits as their phenotype, which I'll talk about in a moment. Mendel studied 7 different plant traits. That's 7 different traits on his 27,000 or whatever different plants. He crossed plants with each other and he studied what their offspring came. So, long story short, he grew a bunch of peas, crossed their pollen, and saw what came out. So he had what he called true breeding plants. These are plants that if they self-pollinate, they will always make copies of themselves. So if you have a big tall plant, it's always going to make tall plants with its offspring. So what Mendel did is he would take plants of one trait and cross them with another trait. Through this process, what we call cross-pollination. The way he did that is he had this little tiny paintbrush and he would literally go and paint pollen from one flower to the next and then snip the pollen off of the flower so they could not self-pollinate. Talk about a boring job. The P generation is what we call the parental generation. The F1 generation, what we call the first filial generation. You can see it illustrated here on the right. There is my P generation, F1. And then after that would be the F2 or the second filial generation. Any crosses between parents with different traits are what we call hybrids. So if they have 2 different traits, we call that a hybrid. Here are the 7 traits that Mendel studied. He studied seed color, seed shape, pod color, flower color, stem length, and flower position. Now let's take a moment and think about that. This is before Excel spreadsheets and all that. This old dude was out there with like a scroll and like some ink, and he was copying down 7 different traits for 27,000 different plants. This dude literally had volumes on peas. That's crazy. So, what did he learn from studying all these peas? Well, the first thing that he learned is that there is what he called a physical particle that is passing information from one generation to the next. He never used the word gene. He never knew what DNA was, but he recognized that there was a physical particle being passed between generations. Today, we call that a gene. No, not genes like that, but genes like a small segment of a chromosome that codes for a specific protein. He also recognized that there was different variations of those particles. Today, we call those alleles. Alleles are simply different versions of the gene. We've been using the analogy that a chromosome is like a book. A gene is a chapter within that book. An allele is different versions of that same chapter. So assuming this is a recipe book, this could be the chapter on cakes. This allele would say make a chocolate cake, whereas this allele would say make an angel food cake, my absolute favorite. Second conclusion that Mendel came up with was the principle of dominance. This is the idea that some alleles are more dominant over others. Now, what this doesn't mean, and I think there's a common misconception here, is that dominance means that's the only trait it is expressed. That is not true. The proteins for the recessive trait are also expressed. It's just that the dominant phenotype is the one that we actually see. But you're making proteins for the recessive allele as well. The third thing he discovered is conclusion he came to is that the idea of that things are segregated, that both alleles from a parent are not separate or not sent to the offspring, so that each parent only contributes one allele to the offspring through the process of meiosis. That through the process of producing pollen or eggs, you're cutting everything in half. So again, that the alleles in the F1 plants are segregated from each other during gamete formation. So he came to this idea of meiosis well before the idea of meiosis was actually discovered. What Mendel noticed was that the reappearance of traits occurred in the F2 generation. So if I crossed a tall plant with a tall plant, but they were both hybrids, what I would get is this 3-to-1 ratio. So we have 3 plants that are the dominant phenotype and 1 plant that is the recessive phenotype. This illustrated to Mendel that these green or parental green plants here actually carried the gene for producing white pods. So that tells us that at some point the alleles that are carried there must be segregated, that the green is being masked or is masking the white. Here's another great illustration just to show you this again. Here is our hybrids. We have heterozygous, and we have the separation, and then it's showing all of the possible combinations that we can have in the F2 generation. So let's talk about Punnett squares real quick and probability. Again, I don't want you to know Punnett squares just to know Punnett squares. I want you to understand that they represent a probability of an event happening. So probability is simply the likelihood that an event will occur. It is not predicting an outcome. So let me say that again. It is only the likelihood that a particular event will occur. It is not predicting the outcome. So what that means is if you do a genetic cross in a Punnett square and you get one organism or one offspring that is homozygous dominant here, that doesn't mean the other 3, if you have 4 children, have to be like this. Every event is independent of the other. So Punnett squares, this is where we use the genes, the alleles, from the adults to combine and make a prediction of what the possible genotypes and phenotypes of the offspring will be. So what is the probability of producing an individual with the mother's phenotype in this cross below? Here we have the mother's phenotype. What would the probability be? Well, this one would be big B, little b. This one would be little b, little b. This one would be big B, little b. And this one would be little b, little b. So the probability would be about 1/2 or 50%. The capital letter represents the dominant allele. The lowercase letter represents the recessive allele. Now, I would encourage when you are choosing letters, do not choose letters that look the same as big capital letters and lowercase letters. So like O, capital O, little o, they look the same. Don't use P, capital P, little p, they kind of look the same. Use things like T, like R, like B, like A, like E. Those are kind of some of my faves. D, sometimes I'm in kind of a D mood. So, tall in this case and short. Gametes are produced by the F1 parents are shown along the top. So these represent the gametes of the individuals. And that's all that's on that slide. Oh, I guess that's cool. Homozygous. Homozygous, let's break it down. Homo, prefix means same. Zygous is referring to the zygote. So what this means is that the zygote has 2 alleles that are exactly the same. So organisms that have 2 identical alleles in a particular trait is what we call homozygotes or homozygous. There are 2 flavors of homozygous. There's homozygous dominant and homozygous recessive, depending on which you have 2 copies of. The opposite of that then obviously is heterozygous, which means 2 different alleles in that zygote. Homozygous are what we call true breeding. And this makes sense. If you allow a homozygote to self-pollinate or to self-reproduce, there is no choice but to pass on only that dominant or only that recessive allele. So all of the offspring will always be the same. They'll be true breeding. However, those heterozygous individuals are what we call hybrids because they can pass on either the dominant or the recessive allele. Cool. Other terms you need to know, you need to go phenotype. I introduced that to you a long time ago. If you don't remember how to remember it, this is how you do it. You want to get physical, phenotype. You want to get physical, phenotype. That's right, there it is. You'll never unsee that. Now, genotype is the type of genes that you have. So physical characteristics is the phenotype. The genotype is the genetic makeup. In this case, we have the phenotype is red, but the genotype is, we are not sure. Because in this case, we don't know if this red is homozygous or heterozygous. So that second allele, it could be either. We don't know. But we could find that out by crossing it with a recessive true breeding flower. So in that case, that's what we would do. We would have to cross it with a flower to determine what the other allele is. Here's a little comedy break for you. There's some heterozygotes. All right, back to the game. The plants have different genotypes, but can have the same phenotype. So what that means is that we can have a homozygote tall, or we can have a heterozygous tall. They both have that dominant allele that is expressed as the phenotype. That doesn't mean that this one is not making the protein for the recessive allele. It just simply means that that phenotype is masked by the dominant protein. So, probabilities predict averages. What this means is that you can't just do this off of one outcome. You can't predict the future off of a sample size of one. And that is why old boy Mendel grew 27,000 pea plants. Like, the dude had a serious data set. So he could make some of these predictions. In genetics, obviously that means we need to have large sample sizes to make those predictions. And those predictions are never going to be 100% accurate because, well, they're just a prediction, and there's lots of other things that are in play. But the larger the sample size, the closer to reality we'll be able to predict. And that's all I have for you today. I know that was quick. I know that was sweet. But I hope you learned a little bit about old boy Gregor Mendel, the brother who's the father of modern genetics. Here is your moment of monkey. Enjoy.